Hello, everyone, and thank you for coming to my talk on the evolution of mannequin vision. So mannequins are not the most speciose group of birds, but there's a lot of diversity in the uh, plumage and behavior of mannequins. And recently, the Mannequin RCN has sequenced 6 mannequin genomes, illustrated by the arrows here, across the mannequin phylogeny in both subfamilies of mannequins as well, the Piperinae, aka the fancy mannequins, and the Neopelminae. So in this branch leading to the Piperinae mannequins, which diverged from Neopelminae about 16 million years ago, mannequins underwent a series of changes, including a transition from a mostly insectivorous diet to a more frugivorous-based diet, a transition from mostly drab, green, olive, brown plumage colors to the bold blue, yellow, black plumage colors we know in male mannequins today, and as well as a transition from more rudimentary displays to the highly athletic and acrobatic dancing displays that mannequins are so famous for. So how did the visual phototransduction pathway evolve with these changes? So the visual transduction pathway is a long protein cascade that starts with light coming into the eye and ends with the brain producing an image to create vision. And the pathway starts with the opsin gene family, the opsin genes, which I'm interested in learning about the evolution of specifically. So the opsins are a group of 7 transmembrane domain G protein coupled receptors, and the 296th amino acid in the opsins is a lysine that is bound to retinol, which is illustrated in purple here. And retinol sits in the middle, in the pocket of the opsin. And when light hits retinol, it causes a conformational change in the retinol, which subsequently causes a conformational change in the opsin, which initiates this phototransduction cascade. So if you listen to Chris Balakrishna's talk, you know that we have, from our mannequin genomes, we have a lot of alignments of genes. We have thousands of gene alignments that were made in an automated fashion using this tool called coats that we used. And a limitation of coats, which is actually a positive in many respects, but the limitation here is that coats will throw out a gene if one taxa in your alignment in your data set does not have the gene. It will also throw out regions of the gene that are not present in all taxa. So a gene may be thrown out and be missing, or it may be truncated. But for our purposes, we only needed an opsin from Neopelma and at least one fancy mannequin to make some sort of comparison or statement about the evolution of the opsin. So we went into the mannequin genomes as well as a variety of other bird outgroups to obtain the bird opsins. So these are the bird opsins listed here, and there's different classes of opsins, including the visual opsins, such as RH1, which is in the rods involved in night vision, as well as the 4 cone color detecting opsins. There's the melanopsins, which are involved in non-visual light responses like attraction or avoidance, for example. There's the non-visual opsins, which are expressed in a variety of tissues. There's the pineal opsins, which are involved in, largely in circadian rhythms, and the photoisomerases, which are opsins that are involved in converting that retinol back into a form that the visual opsins can use. So we were able to recover a subset of these opsins. We missed 4 of them. And in the end, we had a final data set of 11 different opsins from the mannequin and bird outgroups. So we ran positive selection tests using branch sites methods on these opsins. And briefly, we ran AbsRel in HiFi and CodaMel in PAML. And how these work is you input an alignment and you input a tree. And you highlight branches in the tree that you're interested in looking at. So in this example, we have a short alignment here. The branch that I've highlighted is the branch leading to Chirexifia here. And there is a non-synonymous substitution where this blue has changed to the orange protein. And AbsRel would be able to tell us that while most sites have experienced either no substitutions or synonymous substitutions, we have a minority of sites experiencing a non-synonymous substitution here in the branch leading to Chirexifia. And CodaMel could tell us as well that Chirexipia is experiencing some level of positive selection. And it would also be able to tell us what site this residue specifically has experienced positive selection. So for our tree that we put in for our analysis, we have these outgroups, which I mentioned. We have some non-passerines, as well as a variety of passerines. We also have another non-mannequin sub-opsin and the mannequins. And the branches that we were interested in looking at in orange here is the branch leading to all of Pipridae, as well as the blue, the branch leading to Pipridae, the fancy mannequins. So most results look like this. Most opsins experienced purifying selection. So in this example, this is rhodopsin, the rod opsin, and we're looking at the branch leading to Pipridae, the fancy mannequins here. And this is the gene tree. And what you can look at in this gene tree is that all of the branches are thin. There's no thick branches here. They're all the same width, which indicates that there was no positive selection detected. And when you look at the sites on this Pipridae branch specifically, all of the sites, almost 100%, if not 100% of the sites, are under purifying selection. There's no positive selection here. This gene is experiencing very little non-synonymous divergence here and is being preserved. However, there were some exceptions to this purifying selection. OPNM4, the melanopsin, involved in non-visual light responses, experienced positive selection in the branch leading to Pipridae. So here is a thick branch leading to Neopelma and the rest of Pipridae. And although most sites are experiencing positive selection on this branch, a minority of sites are experiencing a positive selection and non-synonymous substitution. So CodeML detected specific sites experiencing positive selection, and this is a long protein of around 500 amino acids, yet 4 out of the 5 total sites that CodeML detected to potentially be under positive selection were within a short, relatively short, roughly 15 amino acid window here in the cytoplasmic C-terminal region of the protein. Now the G protein binds to the cytoplasmic intracellular, the intracellular loops, but the C-terminus cytoplasmic region will bind to proteins involved in augmenting the melanopsin or G protein response. It can desensitize it, cause it to become more active, recruit other proteins, recruit even lipid rafts, do all sorts of things. So this could be a potentially interesting region to show positive selection. SWS1, shortwave sensitive 1, also experienced a positive selection on the branch leading to Pipridae, the fancy mannequins, again, mostly dominated by purifying selection and low omega values, but with a small minority of sites experiencing positive selection. SWS1 is involved in the detection of purple and ultraviolet, excuse me, not blue, purple and ultraviolet wavelengths. And there was one site detected by CodeML to be experiencing positive selection, and that's this site residue 158, which in bird outgroups as well as Neopelma was a valine, but this A has changed to a G in the fancy mannequins to create an isoleucine. And that residue is located in the 4th transmembrane domain of the SWS1. And unlike these residues highlighted in purple here, this residue experiencing positive selection that we detected is not previously characterized to be a spectral tuning site. So spectral tuning sites are residues in the opsin that change the optimal wavelength to excite or create optimal wavelength absorption by the retinol to create the conformational change in the opsin and signal that phototransduction cascade. So if you mutate a spectral tuning site, the wavelength or color of light that will activate that opsin changes. So this site was not previously described to be a spectral tuning site in SWS1. So in conclusion, in the evolution of mannequins, most opsins are under purifying selection, but the melanopsin OPM4 experienced positive selection early in Pipridae divergence. And could this be an adaptation to the low light levels in the forest understory where many mannequins are found? Many mannequins are displaying lower in the forest where there's low light penetrance, and perhaps this elicited or in concert there was positive selection of OPNM4. And for SWS1 positive selection during Pipridae divergence, perhaps this helped detect elaborate displays, new plumage, bold plumage colors that were evolving, or the detection of fruit during foraging. And functional experiments in the laboratory would be needed to detect whether the site under positive selection at the 158th amino acid of SWS1 is a spectral tuning site. You can go into the lab, mutate this site, and determine whether the optimal wavelength absorption of SWS1 is altered, in which case this would be considered a new novel spectral tuning site. So I just wanted to thank NSF for funding this work, as well as the Mannequin Genomics RCN team, particularly Ma Baldwin, Alice Boyle, Elsie Shogan, Emily Duval, and Mike Braun, who helped sequence these genomes, as well as many others. Thank you.